Hi, I'm Betsy Pierson. I'm a member of the faculty in the Department of Horticultural Sciences at Texas A&M University, and my specialty is plant microbe interactions. I'm a newcomer to the field of potato zebra chip. We started some experiments just a year ago with the hope of developing better ways to detect the pathogen. I'm working with Dr. Dennis Gross, who's developing better PCR primers for detection, but our part of the project right now involves when and where to sample. And so we're very interested in how the pathogen moves through the plant. As you know, infection starts with the psyllid infecting the leaf by injecting the pathogen into the phloem. And at that point, the bacteria we think is put into the phloem tissue. And one of the things that we find is that when we have infectious psyllids in the field, it's very difficult to detect that the plants actually have the zebra chip pathogen until we get all the way to harvest and we find that the potatoes have the symptoms. We're looking at the pattern of bacterial translocation in the plant. And what we're doing is forcing the psyllid to feed on middle tier leaves of both susceptible and tolerant plants, and we're then looking at how the pathogen moves through the plant. And we've had some very interesting findings so far, and one of these is the finding that after feeding, you can't detect via PCR the pathogen's presence, although it is present in the plant. When we first are able to detect the pathogen, we find that it moves in a very obvious way, and that is that it moves from source to sink tissue. So if we infect a middle tier branch, the pathogen will tend to move upward to the new leaves and then eventually spread downward through the plant and eventually infect other stems. This has important implications for sampling for the pathogen in the field. If you understand how source to sink movement in the phloem takes place, it gives you a little bit better idea of where and when to look for the pathogen. So that's one of the things that we're doing. Eventually, what we hope to do is look at the differences in disease development in potatoes that have proven to be tolerant via screenings by the Texas Potato Breeding Program run by Creighton Miller. Look at disease development in tolerant potatoes versus susceptible potatoes and ask the question, are there differences in how the pathogen translocates and how the symptoms progress? And if there are, and we can characterize those, could we come up with selectable markers for improved tolerance breeding? So future research will focus on using fluorescent in situ hybridization to be able to visualize where the pathogen is and how it's interacting with the host tissue, as I said, in susceptible and tolerant plants. And we're just really excited to be a newcomer to this program. We're very happy that AgriLife Research has given us an opportunity to start working on the very, very interesting problem of zebra chips.